Okay, how's it going, guys? I'm at home in the kitchen here, and I thought I'd do a home video with you. As you can see, I'm feeding. You can see the scar over there. The surgery was a little rough, but I survived. And it's a lovely evening. The sun is about to set over there. I might show you the sunset if we go on that long. I doubt it, but we'll see. Anyways, well, I thought I would go back over with you real quick. What happens with osmosis. The special case of diffusion where we have movement of water across a membrane from a region of high concentration to a region of lower concentration of water. And this is classically demonstrated with the famous red blood cells. As you can see depicted in this black and white diagram anyways, these are supposed to be red blood cells. Notice how they're kind of biconcave disc. They're lifesaver shaped, if you will. And this is the normal shape of the red blood cell. Biconcave disc, about 7 microns in diameter, and the middle goes in. Concave, well, since I am home in the kitchen, it kind of makes it easier to show you what I mean by concave and convex. This is an ordinary teaspoon, right? Now you notice two sides. This is curved up. That's convex, curved up. It kind of like magnifies almost a little. You can see the camera on the back and a couple of stickers on the back. This way, it's concave. When you eat with it, it's concave. There's a little cave and there's a little depression. So what we have here with the red blood cells is biconcave. The red blood cell is concaved in on the top. When you flip it over, it's still concave on the other side. No, there's no convex side associated with it. Ordinary spoon. If you get confused about concave and convex, you can see the camera in my hand in the reflection. This way it kind of magnifies, and this way, I don't know, it makes it a little smaller, I guess. Okay, well anyways, back to the topic at hand. When we take the red blood cells and we put them in salt solution, salt solution would be over here. Cells placed in concentrated, we do concentration in classroom today, means there's lots of salt, they shrink up. They shrivel or shrink up. That's what this is supposed to be showing you with the evaginations here, the projections coming out. The cells themselves are shrinking up. Water goes out. See the water? Water molecules go out. If we put them in pure water, distilled water, which is 100% water, they swell up and they'll even burst. So here we're putting them in lots of salt, hypertonic solution, they shrivel up. Hypotonic solution, no salt, no sugar, no pure water. The water goes in. Water always goes from where its concentration is high to where its concentration is lower. So water goes, in this case, water goes out to try to equalize, make the salty water less salty. Here, the water goes in. It tries to make the distilled water less distilled, if you will. Hence, the cell eventually ruptures because it can't achieve that. And you can see it here again with this other model. We use this other diagram. There's the famous red blood cell, biconcave disc. Here, these dots out here are representing salt, sugar, whatever. And if we have lots of them outside, hypertonic, the cell shrivels up. And so I hope you got that. We're up to 4 minutes already. I just thought I'd throw this quickie in for the 8th graders. Introduction to photosynthesis. Remember, boys and girls, here is the plant. What does it need to do photosynthesis? Sunlight and carbon dioxide. That's actually pronounced correctly. Carbon dioxide. And from the roots, it gets water. Carbon dioxide and water, and it makes sugar and gives off oxygen gas. It also takes in some minerals. The plants do require some minerals, but the minerals are not food. Its food is the sugar. Once it makes that sugar, it can use the sugar to do respiration, make carbon dioxide and water again, and release the energy in the form of ATP molecules. Well, anyways, I'm almost up to 5 minutes. I better go. Nice chatting with you again. Bye-bye.